I think we'll go ahead and get started. Good evening, everybody. Good afternoon for or wherever you are. Um, Welcome to the Phase 3 town hall for DNA. I'm going to start out with some introductions, just to remind everybody who is on the the ASCA DNA Executive Committee. That would be Ann McCabe, our second vice president, Jillian Ward, who is in, she's traveling right now. She may be able to join us later, but we'll see. Um, and then we have Liz Biscuits, who is one of our technical experts. And we have Peter Hellmeister in Europe, who's one of our technical experts. And I'm Chuck Klein, your first vice president. So, what we want to do today is recap our phase 1 and 2. We want to talk briefly about the Orbit data transfer, and then we're really the main part of the meeting is to have Lisa Gershoni from VGL make a presentation to you guys and kind of give you an update on where we are with that. So let me get started with some of the phase 1 and 2 recap. The board is thrilled to have entered into an affiliate agreement with Veterinary Genetics Laboratory, VGL, at UC Davis. And they will provide ASCA DNA parentage services. The technology will be the SNP technology, or SNP, which stands for single nucleotide polymorphism. And it will employ the ISAG, which is the International Society for Animal Genetics. The gold standard markers will be used for determining DNA parentage. VGL is considered the leading animal DNA parentage verification laboratory in the world, which will give ASCA the accuracy, stability, and longevity, needed to ensure the program's success. And we're really excited to be partnering with VGL on this because of, we know that they're not going anywhere. They're going to be around. They're stable, and they are one of the leaders in DNA parentage. So if we recap what we went through for phase 1 and phase 2, uh, phase 1 was dedicated to education with an emphasis on resolving outstanding ORVET cases. We encouraged the ASCA membership to continue using ORVET until the VGA framework was established. And as our time with ORVET is now at a close, we are encouraging members to wait until VGL is ready to start doing the testing. And Lisa will have more information on that in a bit. So phase 2, we discussed the PPG data being unusable due to being proprietary to PPG. And this, we know, the proprietary markers, there's only one in common between the ISAG 2020 and what PPG was doing. So those data will not be transferred to VGL. And then we also discussed that VP status can only be obtained moving forward if VGL has the ISAG marker data on both dam and sire. And then all dogs previously DNA'd will not lose their status within the ASCA portal. But again, if you want to verify your dog's DNA and obtain VP status, VGL requires the dam and sire's ISAG SNP panel. And we understand the importance regarding VP status of your dogs, and of those that were done with other labs or previous labs, and we're working on a solution within ASCA to address them in-house. And we're also continuing our relationship with Certigen in Europe. So they've previously tested dogs with both the PPG and ISAG markers. ISAG was done on their own, and so we have updated our MOU with Certigen, and if your dog was tested, they have the S&P data, the ISAG markers. Those can be sent to VGL to be put in as a foreign entry. And we're also looking at potential options for Canada, but we haven't found a lab up there yet that uses the ISAG 2020 markers. So if you know of a lab, if you're in Canada, you know of a lab, please let us know. So that's kind of quickly the intro and covering phase 1 and 2. As far as the data transfer from Orvet, we've had one data transfer. We've gone through. There was some missing information, mainly due to dog numbers or people's email addresses, and so those have all been corrected. VGL has those, and Jillian Ward and myself will be meeting with Lisa and the head of IT at VGL tomorrow to kind of talk about what's our next step as far as getting that data incorporated into their system. And then the other thing, Orvet is still owes us a data transfer, and they're working on that now. So we should hopefully get that sometime by the end of this month. And then we will go through that, review it, make sure everything is there, and VGL will have it. And not all is lost if you do not get your data transferred to VGL in these two data transfers. If you get that information from Orvet after the last data transfer, that can be sent to VGL and entered as a foreign entry for a fee. So you're okay there. Today, for the meeting, we probably need to keep it about an hour because Lisa has a hard stop. So want to try to keep things moving. If you have any questions during her presentation, about her presentation, or anything else, please send them to the chat, and Anne will go through those and then read them at the end, and we will address those. So let me introduce Lisa. Lisa is a veterinarian and geneticist who specializes in the study of heritable diseases and genomics in companion animals. She completed her PhD at the University of California, Davis in 2019, and her doctoral research focused on uncovering genetic markers associated with autoimmune diseases in dogs. She currently serves as a geneticist and outreach coordinator at the UC Davis Veterinary Genetics Lab, providing education and support in animal genetics to VGL clients and the community. So I'd like to thank Lisa for taking time out to present to you guys. And she should have some really up-to-date information. So we look forward to hearing it. So thanks, Lisa. Take it away, please.
Yes, thank you, Chuck. Hi, everyone. Happy to be here. Happy to answer any questions you may have as well. I'll just have a short presentation, try to go over some of what I imagine are some of the questions you may have, but then happy to take any additional questions. Let me just, I gotta share the screen first. And hopefully I am sharing the correct one.
Yep.
Is that correct? Okay,
yep,
great.
That's it.
Then I'll go ahead and move my camera over here so you're I'm actually talking to you and not looking to the side. All right, so a brief introduction about the the outline of the talk. I'll talk a little bit, just really quickly about, who we are at the VGL, what type of testing we're gonna have to offer for ASCA members. We're gonna talk about how to order a test. And then I'll talk about foreign types. So if you have data to transfer from ORVET that didn't come through the bulk transfer, I'll have some information about that. And like I said, allow some time for questions and answers from you today. So brief introduction about the VGL. You may already know this, but we are a not-for-profit laboratory, and we're part of the School of Veterinary Medicine, but we are a self-supporting unit. We were established in 1960, and the goal was actually cattle producers reached out to us and asked, can you help us? There's been a lot of cheating, and we want to make sure that we want to prevent people from cheating. Basically, we want to guarantee lineage. Is there something you can do for us? And we could, at the time, of course, DNA wasn't available yet, but there was a way to verify parentage through blood protein markers. And so we were using blood protein markers to determine parentage for cattle. Just a year later, we got a very large contract client with the American Quarter Horse Association, who is still with us. And we got other horse clients also for parentage. And then over the years, we now offer parentage testing for over 20 different animal species, including several livestock species, sheep, goats, pigs. We also offer testing for some bison, elk, and various other species. So in the 1990s, with the advent of DNA, we were able to actually start offering DNA testing or parentage testing through DNA, which is even more accurate than using blood proteins. And we were also able to start offering tests for specific traits and disorders as those became available, as those became known. In 2020, we were accredited by ANAB. This is a very large organization that ensures that protocols are being followed and met, and guarantees accuracy and quality of the results produced. So every year they come and they check on us, and they inspect our protocols or testing process, and we are proud to say we've had continued accreditation. We just celebrated our 5-year anniversary last year, going on 6 now, and we're very proud of that. So our continued accreditation means they have verified we continue to follow the high standards of quality and testing. So as I mentioned, we offer tests for over 20 different species. The majority of the species will have parentage verification. We offer disease and other phenotypic trait testing for some of the species, such as you know, dogs, cats, cattle, horses, some for goats and sheep as well. And we actually just launched our first diagnostic test for pigs 2 weeks ago, which was exciting. All of our tests are direct tests. So you also may or may not have heard of this. There are basically 2 types of tests. You have direct tests and indirect tests. So direct tests are tests that are developed to identify the exact genetic variant or genetic mutation, however you want to call it, that is associated with the trait or with the disease. And indirect tests are actually looking at a nearby region to infer whether or not the actual causative variant is present. So as that may already hint at, direct tests tend to be very accurate, highly accurate, and indirect tests are very accurate, but sometimes they do make mistakes. So we always focus on direct tests because we are committed to the highest accuracy in our testing. So tests that will be available for ASCA members, we're going to have genetic markers, which is a DNA profile, and parentage verification using SNPs. As Chuck mentioned, these are single nucleotide polymorphisms. It's a mouthful to say it's basically 1 letter change in the DNA. So, you have the DNA is written in basically 4 letters, ACTG. So at a given position, you can any given dog will have either an A or a C, for example, and that's a SNP. That's a location where there's one variation. And of course, we look at many of such locations. We will offer health testing. We already have tests available, but we are putting together a specific panel for the Australian Shepherds. And we have co-color and pattern testing as well. So regarding the parentage verification, so this is the pricing that we're offering specifically for ASCA members. So it'll be $50 per sample. But if you have a litter, if you're submitting a litter of 3 or more puppies, then you get a discount of $5 discount per sample. The SNP panel was specifically designed for dogs using the ASCA approved parentage markers. There are about 228, I believe, of them. But we also added there is actually a little bit more than 350 SNPs that we are going to use in case we need additional SNPs to verify a more complicated case. And then Chuck already mentioned the ISAG is the International Society for Animal Genetics. And this is a group that collects information from several laboratories. It's voluntary. So they get together every other year. And there are workshops and meetings where people come together and develop standards for high quality DNA testing. And so these SNP panels that were developed, they were brought forward as the gold standard for parentage testing in dogs across various breeds. So all of the ASCA dogs will be tested for all the 500 plus SNPs that we have in our chip. You will always get the ISAG plus all the other additional SNPs that we have validated. Oops, I don't know why it's not. Oh, there we go. Sorry, it was a little slow. And then I just thought I would mention a little bit quickly about how parentage testing is done in case you're curious and not familiar with it. So parentage testing, parentage verification is based on the principle of exclusion. And this is true regardless of the technology you're using. So whether you're using SNPs or you're using STRs or you're using some other technology, there's other ways, older ways that you could do it too. Basically, you're using a principle of exclusion. We're going to be using more than the 200 SNPs that are tested, and I'm giving you a very simplified example here. So what we're looking for is for every puppy must inherit at least one copy, or exactly one copy of each marker from mom and one copy from dad. So here we have an example. I believe you can see my cursor. Am I correct? Yeah, okay. So I just listed generically, right, SNP 1 through 9. And then for each row, we have genotypes for each of those locations. For the dam on the first row, then we have our puppy. And then we have 2 possible sires here that we're trying to determine which one is the correct sire. So we can see in SNP 1, the puppy has 2 letter A's. So if this puppy, she or he, got an A from mom and an A from dad. And so we know that mom has an A to give, but sire 1 does not have an A to give. Sire 1 only has B. So this is what we call an exclusion. Sire 2 does have an A, and so that would work. So that is what we call a qualification. So for this one SNP, sire 2 qualifies, sire 1 is excluded. The caveat is mutations can happen, and so we can't just use one single SNP. So we have to look at a bunch of them because maybe there was a random mutation that could have caused that one sire to not match. So we look at various other SNPs, and as you can see, the green is just indicating qualification here. So we can see for SNP 2, puppy 1 has an A and a B. SNP 3, this is actually an exclusion, it's improperly marked. SNP 3 our puppy has two letter A's. One came from mom. One could have come from the other one could have come from sire 1 or sire 2. And so we do that, for all of the variants that we're looking at, all the markers we're looking at. And in this case, we found sire 1, in addition to one I missed there, so has actually 4 exclusions. So with that many mismatches, we are very confident sire 1 is not the sire. Sire 1 is excluded. An exclusion is 100% accurate. A qualification tends to be upwards of 95% accurate as long as you have both parents included in the analysis. And so we always recommend, if you have more than one sire that's possible, always send all of the sires at the same time. That allows us to make sure if we need, for example, if we have a tie, we're able to do additional markers and verify the correct sire that way. And if we don't have the second possible sire, then we're not going to know that there are 2 possibilities. So we always recommend submitting all possible sires at the same time. And this is going to be especially true if you have for example, very closely related sires, like a father and son that both could have had access to the female, because they are going to share a lot of the genetic markers. They're going to have many of them very similar. So having the 2 samples so that we can compare will ensure accuracy in the result. This is an example of what a report, this is actually a GOAT report, if you're able to read that. So we already offer SNP testing for parentage with goats, and this is an example of the report. So you see that the genotypes, or each SNP is listed kind of going across 1, 2, 3, just for simplicity. Of course, there's a key that translates, what exactly this 1, 2, 3 SNP is. But then those would be the genotypes for each particular SNP. When we have a parentage request, this section here under results and interpretation will actually tell you what the result was. So in this case, example goat, the specific number qualifies as an offspring of the dam and the sire. If it's an exclusion, it would say the goat qualifies as an offspring of the dam, but the sire is excluded, or something to that effect. So those would be listed. And then the SNP panel will be available starting May 26th. That's next Tuesday, just after the holiday. This is a firm date. We're ready to go. So May 26th, you'll be able to start ordering your kits. In terms of health testing, we offer individual health tests. It's $55 for the first test, and each additional health test after the first one is $25 on the same animal, same sample. Tests can be added at any time. So if you send us a sample, and this is going to be true for you starting out. So if you send us a sample just for parentage, for example, today, and in 5 years or in 3 years, you decide, oh, actually, I want to know if my dog has this disorder or is carrier. I just want to make sure. And then you can just go into your account and add the test at that point, and we'll use the sample we already have in the laboratory. So you don't need to send in a new sample, unless, of course, there could be situations where you had ordered several tests and we exhausted the sample. In that case, we would reach out to you and ask for a new sample. But in most cases, we're able to use the sample you provided the first time to add many tests to that sample. When you are ordering the same test for 3 or more dogs, same health test, we offer a $5 off discount as well. So that would be $50 if you're just ordering one health test for 3 or more dogs, $50 per dog. And as I mentioned, we're putting together a health panel specifically for ASCA members for the Australian Shepherd. So the panel is going to include collie eye anomaly, PRA, PRCD, hereditary cataracts for the HSF4 gene, CMR1, canine multifocal retinopathy, cone degeneration, degenerative myelopathy, multi-drug sensitivity, MDR1, von Willebrand disease type 1, which is the appropriate for Australian Shepherds, and and then neuronal steroid lipofuscinosis, which is a mouthful, NCL6, also Australian Shepherd type. And then we are, so we already developed the NCL test, but we're finalizing development of the hereditary cobalamin malabsorption. So that one is giving us a little bit of grief. So we're just finishing, trying to wrap up the validation, make sure we have an accurate test to offer you. And as soon as that's finalized, we'll make the panel available to you. So this panel will not yet be available on the 26th because we do still need to do a few things. However, most of these tests are available as individual tests. So if you're interested in, I only care about Collie-eye anomaly right now, you could actually go in and order just Collie-eye anomaly. So that's, and you will be able to do that at any point as well. So the bundle price will be $125 per sample for all of these tests, all of the tests included in the health panel. So if you're ordering 4 or more tests, then it does become a lot cheaper to just get the health panel. In terms of co-color and pattern testing, we do offer a basic co-color panel that should meet the Australian Shepherd breeders' needs, and that will include MC1R, agouti, brown, dilute, dominant black, and white spotting. And we also offer Merle, but Merle is not part of the basic co-color panel. It is a separate test. So the first co-color test is $55. The second co-color test on the same sample is $20, and the third onwards is $15 each. And again, these tests can be added at any time in any combination or as a single test, however you prefer. Fully customizable. The co-color panel is a discounted bundle for those 6 tests for $100 per sample. And then for ASCA members, if you would like to order the basic co-color panel plus Merle, you will get all of those tests for $115. Then how to order testing. So I have this kind of step-by-step list, but instead of doing that, just like I did last time, I'd like to actually show you. Let me see how I can do that proper. Here we go. So I'm just going to log into my personal account. And this is a test site that our IT lead has put together so I could show you because it's not yet available. You go into order test and then dog. And then you have a little checkbox at the very top where it says registry affiliation. So when you check on that, oh, and he didn't enable for me, unfortunately. Let me refresh and see. Well, okay. So this is one we already offer. It will work the same way. We will have just ASC is going to be the file code for the for ASCA dogs, and it's going to say Australian Shepherd ASC. So you'll just check this box, and then you get the discounted rate. I apologize that this didn't work out today. You will fill in your dog's name, sex, and breed. So these are the little indicated in an asterisk are the required fields. If you don't have a registration number, that's okay. You don't have to add that in yet. You can add a microchip number if you have it. It's not required. If you have registration number later, you can add that in later and we can reissue a report for you. That then comes with the registration number. So the minimal information we need is breed, sex, and animal name. We do use sex as a quality check. So we check for genetic sex and what our client has indicated the sex is for the animal as an internal quality check. If you submit a dog that's supposedly a female and it's testing as male, that's a big red flag. We need to make sure that we didn't do anything wrong. So we actually scratch everything and go back and start over. So you will see that once you fill that in, you have the section that says owner information. So it isn't as clear, unfortunately, but this is the address to where the swabs will be sent. So if you have a co-owner or if you have, your puppy is already with their future owners, but you want to swab, you want to test them, you can actually edit the owner information to that address and we'll send the swabs to that address. But the results will be in your account. So then when you select the breed, we'll actually have it, we'll filter the test to what's appropriate for your breed. So you're going to have the SNP test available. It's going to show up right down here. And then you'll have the ability to select genetic markers report without parentage. So this is just a DNA profile. But if you're actually looking for parentage, then you can select parent verification. And that opens up a couple of options for you. If you check the box, verify sire to this animal, then you can go in here and you can search your account for animals that qualify. So in this case, let's see, I have these animals. I have dog 1, dog 2, dog 3. So it might be dog 1 or it might be dog 3. So you can add those two possible sires. If you haven't tested your sires just yet, you can actually add new sire by clicking this box over here and just put in your sire information. You can put in the color is optional and breed. And then this is also optional, registration number and date of birth, although we highly recommend entering date of birth. Then you click Save New Sire, and you're going to go and do the same thing for the dam. Once you're done with that, so you're actually only going to have one option of sample choice. We will send you the cytology brushes. This is because the technology that SNP testing uses is very finicky. So we really need to use our cytology brush for optimal results. And so we'll be sending these. There's no additional charge, and we'll mail these out to you with USPS priority mail. Lastly, if you want and you have information, you can fill in the sire and dam of that animal you're testing, agree to the terms and conditions and our turnaround time. And then if you want, you can check the box that says sample submitted can be used for research purposes. If you do that, you will be contributing to advancing scientific knowledge towards the health and well-being of dogs. And if your sample were to be used, there would be no identifiable information ever shared. So not of you, not of your dog, anything. It's always de-identified information. So if you're interested in that, you can check the box. That's optional. Then you click Save. And in order to be able to show you this, I actually have to fill in the required. So I'm just going to call it test. And then you click Save over here, and it'll take you to a page where it says, where you have the option to order test for a new animal if you'd like, where you'll start the whole process with a new form, or proceed to checkout. And if you look at the very top, there's a little cart icon, and it has the cost. And it says 44 here because currently we only have STR available, and that's kind of what I selected there, so that's why it says that. And then we always ask that you double check, that the name matches, that all the information you entered is correct. If it's not correct, you can scroll down and click Edit, and you can go back to that form and fix any kind of information. If you're ready, you click Proceed to Checkout, and that will take you to a secure payment website. Right. Ooh. How do I move this now? There you go. All right, so back here. Then touching on transferring individual or vet data. So if you didn't make it to the bulk transfer and you have a foreign type, we call them foreign type because they were generated at an external laboratory. We have a $20 fee to enter a foreign type. They do have to be compatible. So if you're not sure, you can send us an email. Let's say it's not from OraVet, it's from a different laboratory. If you send us an email to the customer service email, we can check for you whether the report is compatible. So the SNPs need to be ISAC approved and genotype to ISAC standards. But if you send us that, send us an email, we can write that down for you and you can check with your laboratory or just provide a copy of the report and we'll be happy to check on that for you. Then this is something I didn't touch on last time, but I would really like to talk, just take a 5 minutes to talk about this because it's very, very important. So sample collection is relatively simple, but you'd be surprised at how easy it also is to not get it right. And so having a high quality sample is really important so that we can get good results and fast results to you. And so the first thing we always ask people to do is make sure you wash your hands before sampling your dog. And that's, or especially if you have multiple dogs, and that's to avoid contamination. Then we'll ask that you open the sleeve carefully, but don't open it all the way so that the brushes don't fall out. Just kind of open it about a third of the way, and then avoid touching the bristles, so the brush part itself. So just hold it by the handle. You're gonna place the bristle head in between the dog's gums and cheek. And we ask that you lay your finger on the outside, just supporting it while you're rotating the brush back and forth about 15 seconds. And this was a very, very friendly and willing French bulldog who had a treat. His owner had a treat in her mouth as she was doing this for us. And it was super well behaved. Not every dog is going to be this chill. So at least a 10 seconds will be enough. We are trying to collect cells, not saliva, so we don't want drool. The drool actually washes off the cells from the brushes. So really we want that contact with the cheek and the gum. And then the last part is very, very important. Just you want to wave the brush gently in the air for about 20 seconds to allow it to air dry. So if you don't do that part, what happens is we dogs have bacteria in our mouth, in their mouth, and the bacteria loves humidity. So if it's humid inside when you put the swabs in, then while your swabs are coming to the lab, the bacteria are growing and feeding on the DNA. So it really ends up eating up the DNA, and we don't have anything to test. So very important to air dry the swab. And then we will send you 3 brushes, and we ask that you use all 3 brushes per dog. So you're going to repeat these steps for each brush, and then just put them back in the sleeve. And you don't need to seal it. We actually recommend not sealing it, just to allow airflow, again, to prevent bacterial growth. Then a couple more just tips to ensure a good sample collection. Make sure that the dog hasn't had anything to eat or drink for at least 1 hour prior to collecting the sample. Again, if they're drinking something, sometimes they're washing. We're trying to get cells that are kind of loose in their mouth. So if they eat or drink, they're kind of already cleaning those cells away, so you're going to collect less of it. If you're collecting samples from more than 1 dog, I already touched on this, make sure to sample 1 dog at a time so you don't accidentally mix and match the brushes, and then wash your hands in between so you don't accidentally transfer DNA from 1 dog to the other, to avoid contamination. For puppies, we recommend that you wait until they're at least 3 weeks old. Our experience is that DNA, for some reason, really isn't, we can't get enough DNA when puppies are swabbed earlier than that. I'm not sure why. I don't know that anyone does, but we have a very, very high failure rate when puppies are swabbed younger than 3 weeks. So we recommend at least 3 weeks old. And then we ask that you isolate each puppy from the mommy, from the mom, and from the litter mates, and any shared toys. So remember, we're trying to avoid contamination. So if they play with the same toys and you keep the toys in there, there's still DNA from the litter mates that they might get access to. And for about an hour prior to collecting the sample. And then also avoid nursing for that 1 hour. When puppies are nursing, again, they can get DNA from the mom in their mouth. And so we want to give it enough time after they nurse that gets washed out, and we only collect DNA from the puppy and not from the puppy and the mom. So these are just some pointers, and all of this is on our website. You do not have to memorize any of this. You can go on our website, and we have all these tips and a video and all of that. So that was all the content I had to share. I'd be happy to take any questions.
We had 2 questions come up in
Yeah.
the chat.
Yeah.
One was if people choose to do the Merle test, does it give numbers? Is it output of the numbers?
Absolutely, yes. We do test for the size of the allele and we report the size of the allele. So the numbers are going to be reported, correct?
Got it. And then the kind of two-part question, or two questions. What if they don't own the sire of the litter when I wish for parent verification?
Oh, that's a great question. So all you're going to need is the sire's name and the VGL case number. And I kind of did skip a little bit over that. I apologize. So ASCA will have access to all the animals that have been tested through the affiliation. And so if you can either contact the sire's owner and ask for the VGL case number for that sire, or I guess I don't know how ASCA does that, but I suppose ASCA would have access to it too. And so if they have permission to share, they'd be able to share. So all you're going to need, you don't need to have a sample from the sire. If the sire was tested for SNPs, so that's the other thing. If we did test the sire, but it was STRs, STRs are not compatible to SNPs. They don't work. We can't translate one into the other. So if the sire was tested for SNPs, then you only need the VGL case number and the sire's name. And actually, let me show you where you would do that. Here, so gonna go ahead and create another order. So if we go way down here, so we did parent verification, verify sire to this animal. So you have three options here. You have search my account, enter name, registration, and VGL case number, and add new sire. I showed you the search my account and the add new sire. The one you would need in this case would be the middle one. It says enter name, registration, and VGL case number. So you don't need all three, but you do need at least the animal name and the VGL case number, or at least the registration number and the VGL case number. So you enter those there, and we're going to pull that data, that profile for comparison to your puppy.
Got it. And then the
Great
other
question.
Other question, if one chooses to allow research use of the sample, would there be a need for additional samples to be submitted if additional testing is required in the future?
So we make a point of never depleting a sample. So we would still have sample available. We would not deplete your sample, for research purposes. So you would not, unless you really wanted a lot more testing done. We have not had a situation like that yet. But that's a good question and a fair concern.
Got it. So far those are the questions.
No, there's another one, Ann.
Right, but yeah, it's not a Lisa question, but I can answer it.
Okay.
Yeah, so the question, this is one that's come up a lot, and I can answer it. So question was, in order to get VP status, do we need to sample both the sire and the dam if they previously had CP status with Oravet? If the dog has been tested with Oravet, then that information will be transferred to VGL and any puppies will be able to get VP status. If the sire or dam was tested with a previous company to Oravet, then that is not automatically possible and the sire and dam would need to be retested. And then the follow-up is what happens if the sire or the dam of a current litter has passed? As I said, so if their DNA was done by Oravet, completed by Oravet, and then therefore it will be transferred to VGL, even if a parent of the litter has passed, then they can get VP. But if a sire, or in particular, was tested by a previous lab, then there is no way to get a comparison, and any puppy would be CP when they had their DNA analyzed. So that is the answer to that question.
Thank you. So are there any other questions for Lisa from anybody? Oh, there we go.
What will the turnaround time look like for the DNA testing, and then how long does it take to process through ASCA? So you had said before, Liza, the goal is that, so after the sample is received, because we all love that we can't do much about the post office, so you order a member would order the swabs would be shipped, they would collect the sample, and they would ship it back to Davis. And then what is the turnaround time for Davis?
Yeah, so our turnaround time is at least 15 business days. On average, it's 3 weeks, basically. There could be situations where there would be a little bit of a delay beyond that, but we don't expect there to be a whole lot. So the particularity of SNP testing is samples are tested in groups of 24. And so we need to get 24 samples in to be able to do the testing. The analysis itself can be done fairly quickly. And we will always do our best to release results as quickly as we possibly can. That's our priority.
And am I correct in the understanding that once the results are done, since the member will be associated with ASCA, and the office will have the ASCA login, that pretty much immediately after it's done, it will be available for ASCA to access.
Yes, absolutely. Yeah, that's another good point I forgot to mention. So when you select affiliation for your sample, you get a copy of the result, and ASCA gets a copy of the result immediately. So at the same time, as soon as the result is ready to be reported, it gets reported to both to the owner and to ASCA. The affiliation is case specific, so you will need to select affiliation for each individual dog you would like to affiliate. It doesn't affiliate automatically all of the dogs or all of the animals in your account, so you get to decide on that, but it will default to your previous selection, so the form will remember what you selected last. But it's still, of course, a choice. We do recommend checking the affiliation box to make things simpler and quicker. But you also could, if you have tested, if you have maybe, well, I guess the STRs wouldn't, but if you had an animal that you forgot to select an affiliation for and you would like to affiliate later, you can also do that later.
And then question came up, can we have extra swabs shipped to us to have on hand for future testing to save the time of shipping the swabs to us?
So we do pre-labeled swabs. So what we do recommend is if you have a litter that you're waiting on and you kind of know what the due date is, you can actually go in and pre-order tests ahead of time. And if you don't know what names you're going to be using yet for the puppies, you can use placeholder names. And then once you actually have the names, you can go in and change that in the account and it'll update. So yes, you can, but you would need to place the order ahead of time so we can send you those pre-labeled brushes.
Got it. And then the question which was in the beginning was when we can start ordering, and that
Yes.
so that the date is Tuesday, May 26th.
Correct, right after the holiday.
Yes.
And the reason it's not Monday is the university's closed, and the whole thing is closed. We would not be able to do anything even if we wanted.
Yeah. And then the other question, which I know and either Chuck or Liza can address this, but in terms of so obviously members can open an account on Tuesday because they can start ordering on Tuesday.
Mm-hmm. Yeah.
So that is certainly true. And then the other question is when those existing dogs, and Chuck, this is related to the data transfer, will be in people's VGL account.
We are meeting about that tomorrow, so hopefully we can get you get everyone an update on when their accounts may be created based off of the current data set information that we have from that. So stay tuned on that. Hopefully we can have something out pretty soon.
And just to add to that, so the last meeting we had, there was a question related. So what if you already have a VGL, my VGL account? So if the email, the way that we're going to create accounts is the email associated with your membership is what's going to be used to create an account for you for the transfer data. So if you already have an account using that same email, we're just going to merge the data. So we're just going to transfer it there. If by chance it's a different email, then we can work with ASCA to just confirm that the data belongs to you, and we can actually transfer instead of creating a new one in a different email that is associated with ASCA membership. And if you want it on a separate, then as long as we can confirm ownership to you, then we'll transfer the data directly to the account you want. So we can work, we can be customizable in that sense.
And so then the follow-up question is that, yeah, if they already have an account, it will be merged and then their dogs will just appear in their VGL account.
Correct. Yes. They will have, so because these will be foreign types, they are going to have a slightly different structure. So it's going to be, I believe ASC is the file code. So it'll be ASC slash and a number. And the slash is going to indicate that it's a foreign type versus a sample that was tested in-house.
Got it. And then a good question, because I have played with it, but I don't know that we've let the office know that how will ASCA get the results? And I know the answer to the question, but you can answer, Liza.
Yeah, so ASCA will have a portal and access basically a similar account, but all of the affiliated samples are actually going to show up in that account. So that's going to constitute a database for ASCA, and they'll be able to see all of the affiliated samples, and they'll be able to, manage them, and for whatever they need, they'll be able to export copies of the results if they want. So yeah, a copy of each record is going to be under the ASCA account.
Yeah, I actually have the default, and I think is it still kind of listed as test account?
Let me check on that. It might, but
Yeah.
just because it hasn't been activated officially, but I'll check on that for you.
Yeah, so I have the login, I saw it, and it's just search and it comes up because our code is ASC, and so it'll just give you a whole list of the dogs that have been tested. Yeah, so it's
and all of the foreign types, so all of the ORVET transfer data will also be in ASCA's account.
Yeah.
So we're right up. I know Lisa has a hard stop, so we probably need to stop. Really appreciate you joining us and presenting. I would encourage you guys to send your questions if you have them to the DNA help desk, and if we don't know the answer, we will be in contact with Lisa and get that answer back to you. So
Chuck,
please.
if you're okay, I don't mind answering that last one. It's really
Okay,
quick,
well,
and I can
okay,
show the
yep.
screen. I can share
Okay.
the screen again.
Yep.
So Yeah, so here's the, like I said, this is my personal account. So on the bar, wherever you are, this menu bar, you could do view test results or click on all, and it will load all of the dogs that you have or all of the orders you have in your account. So I have a lot of test orders in here and some real orders down here just because I use this account to, exemplify. But basically, you just click on view test results all, or just, the ones that have results. So all will show you all the orders that you have, even ones you haven't completed yet. And if you click on results, it actually gives you only the ones that already have results for. So your dogs are going
Okay.
to show up here.
Got it.
Okay. Thank you.
Yeah, absolutely.
And we're really excited that we have a date now. So
Yeah, us too.
things can start moving. It's been a long road, but I think we're there. So
Yeah,
that's great news. So again, thank you very much for joining us. And
my pleasure.
um, some of us, or I will be talking to you tomorrow. And thanks everybody else for joining us on the call. And we look forward to the future.
Yeah. All right.
Yep.
Thank you, everyone.
Thank you. Good night,
Thanks.
everyone.
Have
Good
a good
night.
evening. Bye.
Bye.